Hello and welcome to the night before news for Biology Paper 1. In all your science papers, it's really important that you check you have the correct paper in front of you when you start the exam. So for this time, in your mocks, you are looking for Combined Science Trilogy, and you need to make sure that it's paper 2 and that it is foundation tier if you are in sets 2, 3 or 4, and that it is higher tier if you are in set 1. On all the papers there are a range of questions, from multiple choice tick box answers to fill in the blanks where blanks have been left in sentences for you to fill in, to short answer questions worth 2 to 3 marks, long answer questions which will either be 4 to 6 marks. Then you've got data and graph interpretation, and in one or more of your papers you will be asked to plot a graph. You'll also be asked to carry out calculations that are specific to that exam paper, and then you'll also be asked to carry out a percentage of maths on that exam paper. In short answer questions, they're going to expect you to be able to recall key information and facts that you've learnt throughout the study of Unit 2 and use Tier 3 scientific words accurately within your response. So, for example, things like homeostasis, which is the control of the internal body conditions within an optimum range, and things like synapse, which is the junction between two neurons which allows an impulse to be transferred from one neuron to another. Long answer questions will quite often require you to recall a practical procedure, in which case it's fine to bullet point the steps in order, but they might also ask you to explain or describe a process. So in paper 2 biology, that could be something like an evolution question, and again it's okay to bullet point those steps of the process of evolution by natural selection one after the other. So for example, you have variation within a population. This variation, some individuals are better suited to their environment. If they're better suited, they therefore will survive any changes in environmental conditions. If they survive those changes, they can then reproduce. If they get to reproduce, they can pass the genetic information on that codes for that adaptation to their offspring, and over time more of the population will have this beneficial characteristic which allows them to survive. Command words, so describe words, literally ask you to describe usually the patterns that you will see in data or in a graph, and as one variable goes up the other one goes down, for example, or this variable increases to this point on the axis and then after that it remains constant, or it might drop back down again. You're literally just communicating what you can see on the graph. They don't expect an explanation. However, if you see the word explain in the start of a question, then they are expecting you to use scientific knowledge and understanding to state why that change has taken place. So for example, why, or explain why a person's body temperature has gone up after carrying out exercise. Well, you would be wanting to link the fact that they undergoing more muscle contractions, which means that respiration needs to take place at a higher rate, and respiration gives out energy. That energy gets transferred to the blood, so the person's body temperature goes up. And then finally, evaluate questions, which are where they give you multiple sources of information and you have to talk about the pros and the cons of each one and then make a judgment of which one you think most accurately links to the evidence. Understanding and being able to identify the variables in any experiment is also really important. Remember, there's 3 groups or classes. The independent variable, that's the thing in the experiment that you have decided to change, and it will always be in the first column of a table, and that could be something like different temperatures or different pHs or different light intensities, but the key thing there is that you or the person carrying out the experiment has decided to choose those values. Then you've got the dependent variable, and its name is given to the fact that it depends on what the value of the independent variable is, because we're looking to see if there's a link or a correlation between the two variables. And that could be something like time for something to react, that could be the volume of gas produced in a given amount of time, or it could be a color change, for example. And then finally you have control variables. Now these are all the other variables in an experiment that if you allowed them to change, they could affect your dependent variable. Therefore you need to keep them the same. So control variables need to be kept the same. So they could be things like the amount of time you leave something for, the mass of potato chips, for example, in the osmosis experiment, or their surface area, for example. Any one of those things changes, and you don't know if it's your independent variable that caused the change or the fact that you didn't control your control variables. Okay, a quick word on graphing. It's very important when you look at a graph that you understand the scale and what the scale is showing you. So on this example, we've got a section I've highlighted where 10 small squares on our scale is equal to 20 millimeters. Now you need to be able to work out what each individual division or small square is worth. In order to do that, you take the value for 10 squares and divide it by 10. So here it would be 20 millimeters divided by 10, which means every small square has a value of 2 millimeters. That way, when you're reading off the graph, you can accurately give the value that they need. Like all the science papers, they can ask you to recall and apply basic mathematical principles. In paper 2 biology, one of the areas that is specific to it is calculating or trying to estimate the population of organisms within an area. So let me take you through that. First of all, you will be given the dimensions of a quadrat. Now quadrats always have to be measured in meters. So in this example, they've given you a 50 centimeter by 50 centimeter quadrat. So the first thing you need to do is convert that into meters. So there are 100 centimeters in a meter. Therefore we divide 50 by 100. That gives us 0.5 meters. Now to calculate the area of a square or a rectangle, it is base times height. So you just multiply the two lengths together. So that becomes 0.5 times 0.5. So the area of our quadrat is 0.25 meters. They'll also then give you the dimensions of the field or the area which they are sampling in. So in my example here, my field is 100 meters by 150 meters. So I do base times height again, and that will give me 15,000 meters squared. Remember, area is always squared because it's base times height. They'll then give you a set of data from the samples that they took. Now my example only has 4 in an exam paper. There's probably going to be 10 or more. Remember how you calculate a mean. You add all the individual values together. Then you press equals, because we have to abide by BIDMAS, and divide the value, the sum of all of your repeats, by the number of repeats that there were. So in this case it would be 14 plus 9 plus 11 plus 0, then divide the sum of that by 4. That gives us 8.5. Well, we need to round that up because we have to have our mean value to the same number of significant figures as the values that we recorded in our samples. So we're going to round that up, because 5 or above you then round up. So that gives us a mean of 9. So now we've got our area of our quadrat, the area of our field, and the mean number of plants that were counted in our quadrats. To estimate the entire population, we need to then work out how many times does our quadrat actually fit into our field. So in order to do that, we take the area of the field and divide it by the area of the quadrat, and that gives us 60,000. So our quadrat would fit into our field 60,000 times. Once you've got that value, we can then multiply it by the mean number of plants, and that will give us an estimate of our total population of plants in that field area. So that then becomes 60,000 times our mean average of 9, which gives us 540,000 plants in our field. Finally, we'll look at some of the math skills they might ask you on biology paper 1. Now 10% of this paper will just be mathematics that you've been taught in your GCSE maths class, but it won't generally be above a grade 4. So they might ask you to round to a certain number of significant figures. So in my example here, I've got 0.2239. Well, the 0 is not a significant figure because it's 0, so we start from the first value here, which is the first 2. And then we go 3 numbers down, so 2, 2, 3. We're focusing on the 3 here, and what we're looking at is the number directly to the right. If that value is 5 or above, then we round this 3 up. If that value is 4 or below, then we would round that down. So in this example, we round it up to 0.224. Another area they might ask you, especially in the context of the osmosis required practical, is to calculate the percentage change in mass of something. So in order to do that, you work out the change in mass, so how much mass has been gained or lost from the start to the finish of the experiment, and then divide it by whatever the initial mass was, so whatever the starting mass was, and the sum of that you would then multiply by 100, and that would give you your percentage change in mass. And the last thing that is quite specific to biology paper 1 will be surface area to volume ratio. So if you look in the bottom right corner, I've got a cube that is 2 millimeters by 2 millimeters by 2 millimeters. To calculate surface area, well, the area of one of these surfaces is the base times the height, so that becomes 2 times 2. But there are 6 sides to a cube, so we need to multiply the value of one surface by 6. So 2 times 2, then multiply that by 6, gives us a surface area of 24 millimeters squared. Then the volume is much simpler. It just becomes base times height times depth. So that becomes 2 times 2 times 2, which would give us 8 centimeters cubed. Then to work out the ratio of the surface area to volume, you do it in that order. So you take the value for surface area, which is 24, and divide it by the value for volume, which gives you 3. So this actually becomes a ratio of 3 to 1.